Hello, Dr. Sedana. My name is Daniela Herrera, and this is my presentation for exam 2. The first question that I'm going to address is a treatment that has become more common in the female population, and that's Botox. Botox is a treatment for wrinkles and muscle spasms. Botulinum neurotoxin A, it's the scientific name of Botox, and it's produced by the bacteria Clostridium botulinum. There are 7 recognized serotypes, ranging from A to G. This neurotoxin acts at the neuromuscular junction where it binds to the presynaptic cholinergic terminal and inhibits the release of acetylcholine. The effect of this toxin is functional denervation, which causes muscle weakness and temporary atrophy. So what is the effect of Botox and why is it so popular? So it works by blocking the nerves that contract muscles, softening the appearance of wrinkles. I have attached a picture on the left which shows how an action potential causes muscle contraction. Botulinum A induces muscle relaxation by stopping the release of acetylcholine into the synapse of the neuromuscular junction. Therefore, as you can see in the figure, in step 1, there will not be an action potential. Therefore, there is a reduction of action contractility by binding to the presynaptic neural membrane, followed by the internalization and cleavage of different synaptic vesicles and nerve proteins. And then what this causes is that the fusion of the synaptic vesicles to the plasma membrane is interrupted, thereby preventing the neurotransmitter release at the neuromuscular junction. And therefore, steps 2 and 3 do not happen it can range from weeks to months depending on the amount of Botox that is injected in the patient. The second question that I will be addressing is the mechanism by which the fungal toxin Perfildin A affects retrieval of KDEL tag proteins from the cis-Golgi network back to the ER. So what's the role of these toxin known as BFA in the intracellular membrane transport? This fungal microcyclic lactone Perfildin A is an inhibitor of protein trafficking in the endomembrane system of mammalian cells. BFA dissociates coat proteins from the Golgi membranes and causes rapid disassembly of the Golgi complex, and that can be seen in the figure at the right. Retrograde transport. Retrograde vesicular transport from the Golgi to the ER is needed to retrieve resident ER proteins that have escaped from this compartment, and endocytosis can transport proteins from the cell surface to the trans-Golgi network. The trans-Golgi network is a cellular location where the secretory and endocytic pathways converge. Now, how is it that proteins can be identified, and how is it that they can determine where is their final destination? Well, they have retrieval tags. In this case, ER resident proteins contain a retrieval signal, the carboxyl terminal tetrapeptide, lysine, asparagine, glutamine, and leucine, also known as KDEL, which binds to a membrane receptor in the cis-Golgi region and return them to the ER. So how does this happen? This can be seen in the figure on the left. So the proteins and materials are taken by endocytosis. That's the first step. And then they form vesicles that will fuse with early endosomes. These early endosomes mature to form late endosomes and then lysosomes. On the third step, these vesicles flow from the cis-Golgi network to the ER when the conditions are right. However, by the action of BFA, the Golgi apparatus is disintegrated. Thus, KDL cannot bind to a specific receptor in the Golgi, and the receptor-ligand complex is not formed. As a result, such complex is never packed into a transport vesicle for it to return to the ER, which is its last location. Here are my references, and I thank you for your attention.